Good morning. I'm Christopher Tompkins, CEO and CTO of KromaTiD. KromaTiD provides genomic structural analysis for cellular engineering and gene editing. Today, I'd like to introduce you to our platform, Directional Genomic Hybridization, and show you how we are using DGH to track inserts used to edit genomes, measure misrepair of double-strand breaks during editing, and even map the structure of entire genomes in edited single cells. When a genome is edited, there are two mechanisms that lead to editing errors, giving rise to two distinct classes of variants. Misedits are caused by faulty or off-target editing, forming sequence errors such as indels at either the edit site or off-target homologous sites. As sequence errors, misedits are successfully assessed by NGS, PCR, and other sequence-based techniques. Misrepairs, on the other hand, arise when the cell's naturally occurring DNA repair mechanism misalign broken DNA ends, leading to a rearrangement of large chunks of DNA or a structural variant, such as an inversion or translocation. After editing, these structural errors can be heterogeneous and complex, and they can occur at the edit site, between the edit site and some random location in the genome, or even at sites not involving the edit at all, true off-target inversions and translocations. Because of the size of the variants and the complexity of structures possible, sequencing and PCR are typically able to measure a minority, if any, of the structural variants arising in edited cells. Developed originally in conjunction with NASA, DGH is a simple, robust process that has been used for almost a decade to assess DNA damage. DGH is a method that's designed from the ground up to measure complex and heterogeneous structural variation. In short, metaphase chromosomes are prepared by stripping the daughter strands, leaving only 2, not 4, strands of DNA. One parental strand in one chromatid, oriented 5' to 3', the other parental strand physically separated and oppositely oriented in the other chromatid. We then paint these prepared metaphases with tiled hybridization probes and image the resulting fluorescently labeled chromosomes from an individual cell. Analysis of the single pattern allows us to differentiate normal from a rearranged genome, effectively measuring the sequence, location, and orientation of any structural variant in the genome. Let's put DGH into a measurement context. The misedit and misrepair products in a batch of edited cells can range from a single base pair to rearrangements of significant portions of entire chromosomes. Measuring all of these products really requires only two methods: a sequencing method such as NGS and a structural method. DGH is actually a platform. In addition to unbiased measurements or mapping of entire genomes with de novo DGH, targeted DGH can be used to map the structure of edit sites and quantitatively track edit site structural variants. Pinpoint FISH and G-banding are not required and have lower specificity and resolution than DGH assays. They are useful, however, as orthogonal confirmation tests, or in the case of Pinpoint FISH, which uses the DGH probes, for measuring specific structural variants in tissues. Either de novo or targeted DGH methods can be applied to cellular engineering, wherever assessing the structure of an edit site or an entire edited genome is important. There are 3 broad categories of use cases for DGH structural measurements: measuring baseline structural variation before editing to provide a full characterization of the starting material, using DGH measurements to compare and optimize editing processes, in effect using the structure, not just the sequence, to assess the efficacy of an editing process, and as a measure of editing integrity, a structural QC test if you like, demonstrating batch-to-batch robustness of the editing outcomes and control of the editing or insertion processes. A real-world example of how this is applied is in the quantitation of inserts used, for example, in editing or in the production of transgenes. Now, DGH is a simple method, but by cleverly multiplexing different designs, we can generate a very deep and rich data set capable of quantitating any degree of structural damage, complexity, or heterogeneity. In this assay design, for example, we have combined de novo DGH paints with targeted DGH probes to measure 3 effects: rates of random DNA damage arising from handling or cellular stress, insertion rates at a target genomic loci and the two homologs of one chromosome, and off-target insertions occurring anywhere else in the genome. Applying this design to practice, there was little or no random DNA damage observed inserting a 10 kb package into iPSC cells using a viral vector. Insertion, in fact, was quite successful, with about 1/2 of the cells, or 1/4 of the target chromosomes, having an on-target insertion. The structural integrity of the insert, furthermore, was also good, with only one translocation observed involving the target site. However, a little under 10% of the inserts at the target site appeared to be inverted or inserted in the incorrect orientation. Insertion at apparently random off-target sites scattered throughout the genome was also common, with the majority of the cells having multiple off-target integration events. Whether these off-targets are functional insertions, however, is up for debate. DGH imaging data can be quantitative. We assess the signal integrity and locations by examining individual fluorescent channels, measuring the size of individual on-target and off-target integrations. Analyzing this data then allows an assessment of copy number and integration integrity. Diving deeper into two individual cells from the experiment, one having a single on-target and one having two on-targets, we can compare the size of the integration events at the targets to those off-target. This analysis suggests that assuming the off-targets are correct integrations, most off-targets are fragments, averaging approximately 1/2 the size of the on-targets, with relatively few complete or multiple integrations at off-target sites. Tracking 10 kb inserts shows kind of the lower limit of the DGH platform. The upper limit is, of course, whole genome analysis. Shown here is the design of our first whole genome DGH assay. Using 5 colors combined with the size and shape of the chromosomes, this assay can map the entire genome in single cells and detect structural variants as small as 3 kb. This is a true, untargeted, unbiased measurement. These KromaTiD paints are designed to the reference genome, not any specific edit, insert, or variant. This is ultra-high resolution karyotyping for sure, but the ability of DGH to resolve orientation events gives this method exceptional power for measuring sister chromatin exchanges and inversions, variation that usually goes undetected. Because of the density of data in a whole genome image, we often arrange DGH chromosomes in a traditional karyogram view, as shown here for an immortalized leukemia B cell. Analyzing many of these B cell images, we can assess the genome-wide structural integrity of either baseline or edited cells. Cells shown in this example were baseline and unedited, and we wouldn't really expect any measurable structural variants. This cell line, however, appears to be a mosaic, with 1/4 of the cells having lost one copy of chromosome 14, and in fact the line appears to be aneuploidy in 40% of the cells. Sister chromatin exchange events, shown on the left, are not true structural variants, as they self-correct during the next cell division. The rate of SCE events, however, is elevated in some cell types and can be indicative of cell stress and overall repair activity. These types of events are important process control metrics for cellular engineering, but are invisible to most measurements. We report them for every DGH analysis. Finally, the cells shown here are the source cells for the Genome in a Bottle, a DNA source provided by the Correlating Institute that is used as a standard for assessing sequencing methods of all types. As part of our work with NIST Genome Edit Consortium, we assess the structural integrity of this cell line using panels of 5 chromosomes rather than a single whole genome. This cell line is karyotypically normal, but does contain 2 small, previously undetected inversions, one on the X chromosome and one on C19. These do not appear together. The cell line is an apparent mosaic, each half having 1 small inversion. I would like to acknowledge the support of the NHGRI, NIST, and NASA for supporting the development of DGH.